going into somatic hybridization. So for thousands of years, naturally we used to get hybridized distantly related crops in nature only. You have to just depend on pollination. Okay, and we could not hybridize, or we could not combine different species of plants. Okay, so this is where somatic hybridization comes in. Okay, so it involves fusion of in vitro fusion, means in the lab. In vitro fusion of isolated protoplasts to form a hybrid cell, and its development to form a hybrid plant. Okay, so we already did know that protoplast isolation, remember? So you just take one protoplast of species 1 and another protoplast of species 2, and then you fuse them, okay, so that you will get a new hybrid. Okay, so it involves 3 aspects. First, you have to fuse the protoplasts, then you have to select the hybrid cell, then you have to identify the hybrid plants. Okay, because sometimes even if you have fused, okay, when the plant grows, it is not the desired one that you want. You wanted a blue orchid, you got an orange or a rainbow orchid, so you still have to select. So let's look at the first one, which is the fusion of the protoplasts. So protoplast is what? Those cells which don't have cell walls, no? The nucleoplasm and the cytoplasm. Okay, so you fuse them. Okay, so you fuse them by mixing the protoplasts of 2 different genomes, okay, by those methods we have learned, no? Spontaneous, mechanical, or induced fusion methods. So the first method, how to fuse a protoplast, spontaneous fusion, is a natural process which we see in the egg fertilization. Okay, when the enzymes degrade the cell wall, some of the protoplasts may fuse to form homokaryocytes. Okay, so these fused cells may sometimes contain a high number of nuclei, right? So like this is the egg cell and this is the sperm cell, no? So when the wall broke here, for a short time there will be a fusion of the protoplast of the sperm cell and the egg cell. So this is an example of spontaneous fusion. This one, they cannot regenerate into a whole plant, okay, just because the male gamete fused with the egg cell, okay, doesn't mean you will get a new plant. All right, so just because this one from the pollen tube, no, this one, the male gamete and the tube nucleus, just because for a short time it fused, okay, this is the surnage, doesn't mean you'll get a new plant. So the next type is mechanical fusion. So you can just push them together mechanically, okay? You squeeze them together and then you can fuse them by tapping on a slide also. But this can cause damage, okay? This can cause damage. So let's look at the induced fusion. Here we will be using fusion agents, okay? Fusion inducing agents known as fusogens. Okay, these are the chemicals. All right, dextran sulfate, sodium nitrate with a high pH, and calcium, okay, polyethylene glycol. All right, then some of the fusogens is described here in the diagram. Okay, the protoplast of strain A and strain B, you add the fusogen treatment, right? Then the protoplasm, the protoplasm, they will fuse, and then you will have a heterokaryon, means when the nucleus have not fused yet, and then when the nuclei have also fused, you get a hybrid. It's easy enough because we have done protoplast isolation, no? Okay. Then you can treat with sodium nitrate. You can expose them to a mixture of 5.5% sodium nitrate in 10% sucrose solution. Oh, where is the pen? Yeah, there. You fuse them in a mixture of sodium nitrate in sucrose, and you take it for 5 minutes at 35 degrees Celsius. Then you centrifuge. Then the pellet, remember what is pellet, supernatant? I have already explained. The pellet is kept in water bath at this time, for that minutes, and then the fusion will occur. Okay. This treatment has a low frequency of heterokaryon formation when mesophyll protoplasts are fused. Okay. Then when you use high pH and high calcium ion treatment, so we first use in the fusion of tobacco protoplasts. You incubate the protoplast in a solution of 0.4 mole of mannitol, which has 0.05 mole of calcium chloride at this much pH and this much temperature for this much time. It will aggregate and fuse within 10 minutes. So here this protoplast also will fuse. Okay, then next, if you use polyethylene glycol, this one is very successful. Okay, it is used to fuse many plant species because it's successful. So you isolate the protoplasts in the culture medium. You mix them in equal volume of 28 to 56% polyethylene glycol in a tube. It enhances the production of fused protoplasts. Okay, the tube you have to shake and you allow to settle. Then you wash it. Okay, this chemical, it's like that. No, some chemical, it just induces fusion. You don't have to do much. These are the advantages: low toxicity, then high frequency of heterokaryon formation, reduced binucleate heterokaryon. Binucleate means like this. Okay, this is the nucleus. This is also nucleus. The nucleus haven't fused. The protoplast fused, but the nucleus hasn't fused. Okay, then electrofusion. They are placed in the culture vessel with microelectrodes, and then you give electric shock. Okay, we have done this in the previous note, last week's class or last class. It's simple. Okay, they don't show cytotoxic response because cytotoxic means when you use chemicals. No, these chemicals, when they're too much high in concentration, they become toxic to the cell. All right. But this one, the equipment has to be it's very expensive, because it's not just like an ordinary electric shock. It is a very, very much controlled electric voltage. So the equipment is very small, but it's very, very expensive. Okay, because you are shocking cells. You cannot just give electricity however you like. You cannot just shock. So let's look at the mechanism. There are 3 phases: agglutinization, plasma membrane fusion, and then formation of heterokaryons. So first they will adhere together, they will agglutinize. When the protoplasts are in close contact, adhesion will occur. Then plasma membrane should fuse next. Then the plasma membrane get fused at localized cells at the point of adhesion. There will be cytoplasmic bridge between the protoplasts. Okay, cytoplasmic bridge between the protoplasts. Then the plasma membrane fusion can be increased by high pH and calcium, temperature, etc. These are the chemicals we already learned in the we have already learned in the same note. Then formation of heterokaryon. The fused protoplasm get rounded as a result of cytoplasmic bridges, leading to the formation of special homokaryon or heterokaryon. Okay, so when 2 nuclei they occur in the same protoplast, means the fused protoplast. Okay, now the next is to select the hybrid cell. About 20 to 25 of the protoplasts are involved in the fusion. Okay, it's not that just because you added the electric shock or the polyethylene glycol doesn't mean all. Okay, because that is not in the nature. No. Okay, so So there will be a mass, a mixture, no, of fused ones, unfused one. Then sometimes the only homokaryon is there, the protoplast is fused, but the cytoplasm hasn't fused. Sometime, like the organelles, they have behave abnormally. Okay, so you have to select. Okay, so this is the difference between homokaryon and of different types. Okay, so you've fused strain A and strain B. Fine. Here in strain A, no, they have removed the nuclei. Okay, here they want only the fusion of the cytoplasm. They don't want the fusion of the nucleus. So you are getting the heterokaryon with the nucleus of B, but the cytoplasm of A and B. So here neither of the nucleus have fused. Okay, so this is homokaryon of type AA. Okay, means only this of A without nucleus and A cytoplasm. So this is the types that you may get. You may get a cytoplasm of B, and then the nucleus of B. Okay, as well as this one. Okay, this one B. This B has come here. Okay, this one is there, A is there, it's light, I think it's light. Okay, so that is there. Fine, these are the types you may get. Okay, so you can select them through the biochemical method. Okay, means you use chemicals to select them. So how do you select them? These by using the drug sensitivity or auxotrophic mutants, or you can just observe them by visual methods. Okay, so let's look. So if you want to select your hybrid, if you want to use drug sensitivity method, suppose your hybrid is sensitive to one particular medication. Okay, so looking here from the example, it'll be easier. Okay, so petunia of hybrid A, it can form callus on the medium, but it cannot grow if there is this antibiotic actomycin D. Okay, but petunia B, okay, it can live if there is this antibiotic. Okay, so in simple terms, if this is one petri plate, this is another petri plate, we add the antibiotic actinomycin D. Here also I add. Then I grew this petunia A, and here petunia B. I grew here. So what happens? Those protoplasts, those protoplasts or cells, they cannot develop here. They cannot grow. You waited for a week, for a month. Okay, nothing happened. But in B, they grew. The callus formed, the roots and shoots they form. Okay, so that is what it means by resistant.